So what we're going to be doing this afternoon is we're going to start focusing on sample preparation. And I'll just give you a little bit of a primer on considerations for biological samples with a lens more on single particle analysis. And that's what we're going to be doing. We'll talk about grids. That is our sample slide, as it were, and I'll get into a few more analogies. I'll give you a hypothetical idea of what will happen to your sample during vitrification, and we'll touch upon a few newer methods. Tomorrow we'll get to see some of the newer methods from blot-free vitrification, which I'll mention in a little bit, all the way through to some of the focused ion beam sample preparation techniques. In terms of cryoEM, I just want to reiterate what we heard a little bit earlier in the morning. We tend to be quite imprecise in terms of the nomenclature as well as the vocabulary we use. And that's sort of due to the fact that the group of cryoEM technologists was very small. And when we were using shorthand, we knew we were using terms imprecisely, and that was okay. As cryoEM has expanded, it caused some confusion. Joachim highlighted Coulomb potential maps instead of density maps, we know, and resolution, things like that. Cryo-EM, and in terms of what people think of cryo-EM, they most likely are referring to single particle analysis. And that is due to the fact that cryo-EM has three main modalities, and using the same instrument, you can perform all three experiments. So we have single particle approaches, we have electron tomography approaches, and we have electron diffraction approaches. Whether they're cryo or not would reflect the sample preparation and whether or not you're keeping the instrument at liquid nitrogen temperatures. But the root goal for all our sample preparation techniques is to see if we can access as high resolution as possible. And atomic resolution has been used sort of as a proxy because it's very useful as a metric to determine quality, to, by first principles, have validation, and as well as gives you enough buffer cushion to start answering some biological questions. It's always nice to say, I only need 5 angstrom resolution, but I'm at 1. Okay, so I would prefer to have that buffer, and that sort of removes some shadow of a doubt in terms of your analysis. What can happen during the way that we image a sample is that we sort of forget all the steps that are upstream and downstream. And a lot of what's being done in sample preparation won't be fully realized until several different steps downstream. We tend to liken a transmission electron microscope to an inverted light microscope. And the reason for that is that optics for light microscopy really translates really well to electron optics in the sense that we have some sort of source, we have a lot of lens systems, and a transmission electron microscope is very similar to an inverted light microscope in the sense that we're going through our sample, and therefore our sample needs to be thin. And given for single particle approaches, we want to reach the highest possible resolutions. We want the sample to be very thin. There are some peculiarities that I'll get into tomorrow morning about the electron microscope, but we also need that sample to be stable under vacuum. And another way of thinking about that is if we literally inverted a microscope and you compared the lens system, the electron sources, you'll see that it very much goes 1-to-1. So if you're thinking about that, then we have to prepare a biological sample that's amenable for EM analysis. What do we have to consider? Well, the main thing is that I briefly touched upon that we have several different modalities of EM that we can use the same transmission electron microscope with. What we have to be cognizant is that how do we prepare the sample and how do we analyze that? And an electron microscope is just like a microscope. For resolution, you can just go up and mag. But in reality, what ends up happening is that we can't access all resolution regimes with the same sample. What we tend to do is we use different subsets of EM techniques and therefore have to think very hard on what our particular macromolecule of interest may be amenable to. We could have something that's very homogeneous. That's why in terms of other structural sciences, it dovetails really well. If you have samples for, let's say, X-ray crystallography or NMR, they can directly enter in sort of the left half of this pipeline. Yes?
So the ribosome then this morning, like how thick is the ribosome? You know, like is it less than 100 nanometers?
Yes. Most of the biological macromolecules we're dealing with are tens of nanometers typically, or single nanometers, unless you're dealing with really complex phages and other things like that, which can get quite thick. There are other model systems that you might use. Let's say you want to put it in liposomes or other biologic carriers. They can form complexes that are quite thick and maybe too thick for high resolution analysis. And then in that case, you might have to do some combinatorial approaches. Again, underscoring that you might not be able to do one particular sample prep and get everything, whole systems level context as well as atomic resolution.
What's the biggest complex that you hear? It's like you've seen maybe M4, for example. Like what's the biggest at this location that you've seen? Is it under 100 nanometers?
Well, we have collected on things over 100 nanometers, but for single particle approaches, even if you're trying to do single particle approaches on tomography milled samples, then they try to aim around 100 nanometers because you have the best contrast and the best resolution. You can go thicker, and you can use other instruments not here and go thicker. It's just then you would have more moderate resolutions. Okay. So, if you have a 1 micron thick sample, you can still image that. It's just that you won't be accessing this particular resolution regime. It'll be more towards that. And that's why it's very important to also understand what is sufficient. It's always nice to be subatomic, but it might not be resource efficient to get there, nor necessary for your biological question. And the way that we prepare samples, depending on if you're going into cells or you can exogenously just purify and enrich and maybe form arrays, that helps push you into a particular, I guess, sample preparation workflow. Because there are certain membrane proteins, for example, that naturally form 2D arrays on membranes, and that lends itself really well to some electron diffraction techniques. But we'll really touch upon the single particle workflow or the canonical single particle workflow, where typically you can enrich for a macromolecular complex and ideally isolate it in not just structural homogeneity, but identity-wise homogeneity. Typically, how we think about this process is that we purify a macromolecule in solution, and we're going to have to thin it down. What we end up doing is we go from a very low surface area, high volume, to high surface area, low volume. And one way of doing this that you may have seen would be you apply a sample to our slide, as it were, our glass slide in terms of light microscopy for EM would be this 3 millimeter metal mesh. And once you treat that, you can add your sample and you can use a filter paper, common filter paper you find in the lab, to thin out the sample. Normal volumes we're dealing with are on the order of a few microliters. And if you're trying to do that calculation, if you just have a 100 nanometer thick volume that's vitrified over roughly that 3 millimeter metal mesh, you only need an order of magnitude less in terms of volume, actually several orders of magnitude less. But it's very hard to really do a lot of the sample manipulation we want to do reproducibly. And what you can see on the bottom right is that if you have an EM grid, it tends not to be so uniform during your sample prep. Fortunately, as you heard a little bit earlier, we only need a few thousands or tens of thousands of asymmetric units if you have a very well-behaved sample. And there tends to be a lot of real estate that, even though we can't access, there's sufficient amount that we can. And the more reproducible and more experienced or more automated you are in that part in terms of getting good throughput for thin samples across the whole EM grid, the more efficient your whole experiment can be. But you don't have to be 100% perfect, but the more you are, the easier the later steps will be. So you have a very thin sample, you'll put that into your electron microscope, and something that you'll hear a little bit later about is that not only do you want to preserve your sample during the cryo sample prep, you also want to make sure your sample is pretty evenly distributed, as well as has good angular coverage in terms of you have different views of the same macromolecule. Because what we'll be doing will be we'll be taking a lot of images of it, and we'll be doing processing to boost our signal to noise. And as I said, we're not very precise in terms of cryo-EM on our terms, because single particle analysis doesn't mean a single particle. We're averaging multiple particles together, but we're looking at a single target in the whole process. But this is where the enrichment and homogeneity and a lot of the algorithms come in the sense that if we're able to be a good steward of our sample, make sure it's in a biocompatible conformation and we carry it all through the process, then we would be able to start reaching answers to the biological questions we're posing. So let's look at a little bit more on EM grids. There are certain examples of EM grids showing you up here. And I think the hard part now these days is that as we are getting better and better at the algorithms and applying algorithms towards our data, it's very hard to say what a bad grid is. It's very easy to say what a good grid is in the sense that you would know if it's nice and uniform, you have a lot of real estate. But which grid is a bad grid if both examples I'm showing you with a 4-hour data collection, we each got a 2.2 angstrom map, very isotropic. So I don't know what a bad grid is, but I can tell you like this particular grid on the left will take a little bit more than on the right if a microscopist were to see it. But if the end result is the same, then it gets a little bit more confusing. And I think the task would be is to try to be as critical in each of the steps, because again, computation can only do so much, instrumentation can do so much. Sample preparation ends up being very valuable in the throughput and the reproducibility. Something that in terms of academic science, what ends up happening is that we don't tend to have a lot of replicates. When we have a particular platform and we have the result, we move on to the next system. Something that we want to work on is really trying to ensure that we are very robust in the sense that if you have a particular protocol, you can give it to a lab mate, or that can be done later on. And so what we're showing you here is more of an example, this is from cryoEM 101, of not only the EM grid, but overall where we want our sample to be. And I'll get into a little bit more about that grid after we saw the overview of what a grid looks like. In terms of EM grids, we are borrowing a lot from not only material science for single particle analysis workflows, but the room temperature EM crowd, in the sense that we have many different form factors of EM grids, and they're made of a wide variety of materials. Given that we want to be in cryo-EM, we tend to want to have grid type of materials that are non-iron containing, because what ends up happening is that our lenses in the EM are actually magnets. So you don't want to have a magnet in the magnet. And also we want to keep our samples in cryo conditions. So therefore we need something that's thermally conductive to maintain that cold temperature. What's commonly known for grids are copper. A lot of the early grids are made out of copper because it's very malleable, very easy to work with, very easy to control. But there are titanium grids, there are stainless steel grids, there are other types of form factors. We tend to see round grids, but if you see what's being made now, there are square grids, there are hexagonal grids, there are different types of grids. And the reason why we call them grids is that they have a mesh on them. And so it's sort of like these grid squares, and that's what we're referring to a grid. Just to be clear for terminology, when I'm referring to the grid, I'm referring to the metal mesh. When I refer to the foil, I'm referring to what we put on top of the grid, and that ends up being a support film. For negative stain or room temperature work, that could be a continuous layer of carbon, but for cryo applications, there might be holes in that foil. A gold grid to me means the grid is made out of gold. A gold foil gold grid would mean that it's a gold grid that's through and through. Okay, and the reason for that, gold grids have existed for decades because for room temperature work, you would want to culture cells onto grids because copper is cytotoxic. You may hear most people refer to gold grids inferring that both the foil and grid are made out of gold. So just be careful, because if you go to a manufacturer and get gold grids, you might just get that without gold foil. It might be carbon foil, which are also very useful. Something also to keep in mind when we're talking about mesh, these grid squares, the squares you see on the grid, 200 mesh grids don't mean that there are 200 squares on that grid. It's sort of a measure of pitch. So that's 1 inch over the mesh. And what you end up having is that common grids you might see for single particle analysis are on the order of 300 or 400 mesh. Maybe for tomography, you're closer to 200 because you're going to be tilting your sample. You don't want the grid bars to be in the way as you tilt your sample. But yes? Pitch would be, it's relative. So it's a unit over the mesh. That's the pitch. It'll be defined there. So the actual grid square of a 200 mesh, the hole is actually 90 microns. And I don't know how many grid squares you'll have. I think we'll calculate that out. But in this case, mesh in terms of pitch will be 1 inch over whatever, and then that will give you a particular size. Initially, a lot of people came, oh, so there's 200 squares on a 200 mesh grid. No, they're not. It's if you're thinking like, it's very similar to when we're talking about gauges for needles. It's a very distinct type of measurement. It doesn't necessarily correspond if you're not aware. You might not care. A lot of people use a particular consumable, and as long as they're just using the same one, they might not need the details. Here are examples of carbon foil on grids. There are different manufacturers. More often, you'll see regular arrays of holes in our foil. Back in the day when we didn't have the, I guess, the high level of QC that we have now, we were doing something a little bit more homebrew, homemade. That's in panel B. That's called a lacy carbon grid. And those holes were made manually with adding a solvent onto a glass slide. But right now, you probably will see something closer to what's in the panel A or C, where you'll have this regular array. And a lot of data collection platforms make use of that. If you look at a common grid, there's going to be, yes? That was exactly why you wanted lacy carbon grids, is just that because not until recently do you have multiple size holes, you wanted different ice thicknesses across. So people were actively going after lacy carbon grids such that the hole sizes were different and the ice thicknesses were different.
That's something you can control for within one collection. Differences in ice thickness.
Well, that'll be more on the processing side, but you do have some QC in the sense that the way that you make the lacy, you could have a wide distribution or it could be biased to particular sizes, but the irregularity was highly sought after for certain applications. Some people now use multi-size hole grids because they don't really know what their target is. So ideally, if you have a very defined target, it's very straightforward. You have, the full protein sequence and you're good. But what people are going after now are complexes or different types of complexes with different sizes and different conformations. And they are, especially in the early stages, are searching for the most compatible grid. So there are different applications. Most of the single particle work, since the sizes are on the order of a few nanometers or tens of nanometers, if your lab is only stuck using one or two types of grids, that has broad applicability. So you don't necessarily have to have a full gallery of grids if you have a very established system. Okay, so your grid is roughly a couple dozen microns thick. You have a thin foil on top of that may be nanometers in thickness, give or take. And then for single particle applications, we use Holy grids in the sense that there are gaps in the foil, and that's exactly where our sample is. There has been a movement towards gold foil gold grids, and the reason being is that there's been a lot of studies that when you image your grid, that there can be charge buildup and there could be tension buildup. But what ends up happening is that when you cool down your grid and foil, if they're not made out of the same material, they'll have different contraction rates. And what ends up happening is then you could lock in some tension. And when the electron beam hits your grid, you can relieve some of that tension. So by having your foil and grid made out of the same material with the same thermal contraction, it's less likely for you to build up a lot of that force. I don't think anyone has really made use of that. There are mechanosensitive channels and targets. I don't think anyone has been appropriately been able to use this particular byproduct in those particular studies. But what you saw in the video, hopefully, is really outlined in this particular paper, where when they had a metal grid with either a carbon foil or a gold foil, there were different contraction rates. And that also affected the motion of the samples in the grid. Okay, so I'm going to briefly touch upon the vitrification process, and if I have time, on newer methods, but I want to leave you enough time because in about 20 minutes, we're going to be heading downstairs. You saw some synopsis of this in the sense that the way that we prepare samples today is very similar to the devices used in the 70s and 80s. There have been some improvements in terms of automation and cameras, but by and large, what people do today is very similar. What we end up trying to do is we try to work in normal atmosphere and temperatures. But if we're able to cool very fast, somewhere on the order of 10 to the 5 to 10 to the 6 Kelvin per second, we can bypass a lot of the phase transitions of ordered ice to this amorphous water. So it's very much like a glass-like water where it's still sort of a liquid, but in terms of how we treat it, and though we call it ice, we don't really mean ice. We really mean vitrified water, this glassy-like amorphous water. And if we cool that fast, we cool so fast we don't form hydrogen bonds, and we don't add extra strain to it. So there can be the opposite side of this phase transition diagram, because if you heat up to a nice balmy, I guess somewhere around negative 137-ish degrees Celsius, you can go from this amorphous water to then ordered water and crush your sample. So once we vitrify your sample, you have to keep it quite cold. And it's quite easy to warm it up, because if you have a room temperature forcep and you're touching a liquid nitrogen sample, that could be enough to pass through a vitrified state into some sort of order state. I don't necessarily only mean hexagonal ice. When you put water in the freezer, you'll form nice hexagonal ice, and ice, like water, will expand. But there are other types. I think there's 11 or 12 forms of ordered water that may be deleterious not only to your sample, but give high background when you're doing imaging. We usually use a hydrocarbon like ethane or propane, because you'll get the lead-in frost effect if you just dunk something straight into nitrogen, because it'll evaporate and then that'll form a nice insulator. So I mentioned we're going to go from a low surface area, high volume, to a high surface area, low volume. And what ends up happening is then you have your macromolecule in solution, and it's going to bump into what's known as an air-water interface. And in that air-water interface, some deleterious effects can happen. For example, denaturation. There are some advantages like concentration. Something that you might notice is that if you're doing negative stain, you might only need a few micrograms per mil. When you're doing cryo, you might need quite a few mgs per mil. And then when you're doing bulk freezing and then you mill a bulk solution, you might need hundreds of mgs per mil. And so there are good effects and bad effects of the air-water interface. And usually when people are talking about EM readiness, they're hoping that their sample construct won't interact too much with the air-water interface or won't be too bothered by that. And you can do certain things like protect interfaces. You can add a carbon layer or a graphene layer. A couple atomic layers of graphene is very electron transparent. It's like 99.9% electron transparent. You can make a graphene sandwich, etc. But during this process, as you'll find out throughout the day, it can be harrowing. The idea is that you get something like panel A where you have wonderful particle distribution, but even if you have good particle distribution across your grid, you could have several different types of ice, and that could be less efficient for your data collection. For example, in panel F or H, you probably nowadays days can still process through that, but that can lower your efficiency. So the idea for sample preparation is that you get something like in the upper right where you have autolace, where you have that ideal cartoon figure where you have a monolayer of single particles that are not touching to each other in randomly oriented distributions and a very little layer of water. Whereas the reality may be these other cases as you see here. So in terms of making EM ready samples, I think we'll start touching upon that. I think every lab has a particular workflow. There was a mention, when would you do negative stain? If you have a very novel construct, negative stain may be useful. If you're joining a lab that has done 100 years of biochemistry and for the past 10 years has a very stable platform, you may skip not only negative stain, but certain steps of the workflow because it's so tried and true. But every time you're encountering a new scenario, I would say to be very careful and know in the back of your mind what particular pitfalls there are, and such that if you need to do troubleshooting, then you know where to go and how to analyze what may be an EM ready sample or not. Okay, I think I'm running on time. I have all the slides. I'm skipping a few things, but the main thing to note that there was a mention on how thick can you go. If your sample gets thicker and thicker in a TEM, standard techniques are going to be less useful for sample prep. For example, if you were to image a whole E. coli cell, you can put several hundred nanometer thick samples into a TEM. It's just that in terms of the standard sample prep, we wouldn't normally do that because the amount of information we get is quite low. And even if you're dealing with phages that are on the order just shy of 100 nanometers or even 50 nanometers in diameter, there can be quite a loss in terms of the information you can get using single particle methods. Okay. So I'll skip this just to highlight that you'll have these slides. There's a lot in terms of the steps of the workflow. I also have different slides on if you have particular issues, what particular avenues one might try, let's say your preferred orientation or flexibility. A common question would be, what's your favorite buffer? What's your favorite additive? Just like in other structural sciences like X-ray crystallography, there are kits that you can buy commercially as additives. And basically what they've done for you is done a literature search. So if you're dealing with some of these fields or some of these targets, you can do a literature search and see, okay, commonly you can work around certain issues with particular additives or particular treatments. So we do have some of these kits. They are not universal. Very similar to X-ray crystallography, for a lot of sample prep is trial and error. Okay, so